In this second video session, there are 4 main objectives. The first one is to cover off some outstanding issues from last week. The second is to give you some homework. The third is to cover some of the greater complexities that arise in the structure of some of the more highly evolved grass plants, because they can really make species very hard to identify. And I'll try explaining what happens with some of these deviations from the standard rule. And the fourth, and perhaps the most important part, which will occupy most of our time, is learning how to use the identification keys to provide you with a methodical, systematic way of identifying an unknown grass from scratch. So that's what we're going to be doing. I'll start off with the homework, just to get that out of the way, and I'll need to share my screen for that. And yeah. You remember that last time I asked you to read from this lot of notes, the Wild Grasses Southeastern Australia notes, the section in chapter 1. So that has information that is just as easy for you to read as to hear me talk through it. And we need to spend our time together on things that really require me to lead you through things and show you examples and that sort of stuff. So I'm relying on you reading those parts, well, the whole of chapter 1. Chapter 2 begins with stuff that I dealt with last week. Reading through that will help reinforce what I dealt with last week. And I mentioned that I got down to about section 2.5 last week, which is, it's there. Oh, an internal error occurred. That's not what you want to see when you're doing a demonstration. Let's see if I can get it to display properly again. It looks like one page is not going to display properly. Okay, all right. I think that must be the most important page that's gone missing. Can anybody tell me whether page 12 is the start of chapter 3? Let's see. I'll just scroll down further. No, it's further down. Okay, I'm going to rely on you reading section 2.6, which is about the differences that you find between grasses and the similar looking families such as the rushes and sedges and so on. I introduced you to Matt Rush's last Thursday at Toowoomba Vale, and I will be showing you tomorrow and tomorrow week a range of species of rush and sedge. I won't cover a huge number of species, but I'll show you enough to allow you to be more confident at distinguishing a grass from those groups. And if you read page 15 and 16 down to the end of section 2.6, that will help you have greater confidence with that. Section 2.7, I'm going to deal with this afternoon. And after that, we're into chapter 3. And your homework is to read chapter 3 all the way to the end of the notes. Some of it you might find not particularly relevant to you. I'm not going to test you on any of this. It's really just so that you get more benefit out of the time we have together. I will tell you about how to identify grass species, of course, and I'll let you read about taxonomy and how we allocate scientific names to things. Preservation of grass specimens. Now, somebody in their when they filled in the questionnaire about what you hope to get out of the course, somebody mentioned specimens. And I can't remember in exactly what context, but if any of you want to know how to press grass specimens properly to produce a lasting specimen for reference purposes or to lodge at the National Herbarium of Victoria, then I can take my plant press along tomorrow and show you how it's done. Does anybody particularly want me to spend the time on that? Not hearing anybody.
Yes, please, just a little bit. That'd be good.
Okay. Sorry,
Yeah, happy to.
How was that, Rose?
I said yes, please. Just a bit would be good.
All right, I'll try and remember to pack my plant press. Okay, so there's a section there that tells you a bit more about how to preserve or press plant specimens. Ecological variability amongst grass species. I've been covering that and will continue to do so. Then there's stuff about horticulture, including down the end, stuff about revegetation projects. And I'll spend a little time next Wednesday in our video session showing you some examples or photographs of where revegetation's been done and discuss the techniques that have been used successfully for that. But I'm going to rely on those earlier parts of chapter 5 for you to read them because you don't particularly need me to find out those sections, I think. Then there's management. I'll also cover management and show you some examples next Wednesday. And that takes us to the ends of the notes other than the glossary. So I'll leave you to read through that. It might be a matter of you scanning through it and picking up the bits that are relevant to your particular interest. But by reading through them, it'll help you reinforce what I do during the sessions that we have together. These notes are also an enduring resource for you to refresh your memory of all these different aspects that are covered by the notes. That's really important because what you find is over the winter months, we've got more than half of the year when there's no flowering material on grasses. And one tends to forget the important aspects of structure and the terminology in particular, like which are the lemmas and which are the glooms and all those sorts of things. But don't be put off when the grass flowering season starts next year, because although you might not remember the lemmas and the glooms or stuff like that, what people find is that if they get out these notes and refresh themselves as revision, particularly if you can do it in conjunction with a workmate, then you find that it comes back to you. And you can give a bit more practice next year to reinforcing the terminology and the structure and practicing looking at the important bits of a grass plant. And year by year, more and more remains in your memory from one flowering season to another. So just don't despair if you don't remember it all in 12 months' time. You can recover it, and these notes are an important part of being able to recover those lost memories of names and important features. Okay, that's all I need to say about the homework. Has anybody got any questions or comments? No? Okay. Next, let's talk about unfinished business from last week. Has anybody got any questions or comments arising from last week?
Yeah, I had a quick question.
Yes, Keith.
We made a note of the fact that that area had been repastured with a whole bunch of introduced grasses, and I was wondering if there'd been a bit of a push back in the other direction, putting natives back in any sort of meaningful way, or if it's still just a very niche sort of movement at this stage.
Yes, there have been substantial efforts, both in agriculture. I think you're talking about over the fence in the pasture, right? Yeah. And also in places like roadsides. Vic Roads has put quite a lot of funding and effort into investigating, converting weedy roadsides into native grassland in the Western District, for example. I'll show you some photographs of that next time. So Keith, I will explain a bit of this next time, but more from the point of view of nature conservation rather than the pasture. However, there is a book, I don't think I showed you the Native Grasses book, that's the title, by Meredith Mitchell. It's designed specifically for farmers who might be interested in knowing what native grasses they have in their pasture, what ones they might want to encourage. And that book includes all sorts of information about, I think, about 20 different species or genera in the case of wallaby grasses and spear grasses. Information such as the crude protein content and digestibility and how many tons per hectare is produced, all that sort of stuff that a farmer would want to know. And there are plenty of native grasses that are very nutritious, palatable, good tucker from the point of view of crude protein, that that respond well to grazing. Some of them actually increase in proportion within pasture due to grazing, particularly some of the wallaby grasses. And in East Gippsland, I know spear grasses can do that. And there are advantages in those grasses in that they tend to keep going into droughts a lot better than introduced pasture species. So there's a big advantage from the point of view of sustainability in that regard, that you can keep grazing further into a drought and further into the dry periods of each summer. And the native grasses, by definition, they can do without superphosphate and other nutrients being added. So they're much lower inputs. That means inputs in cost and inputs in terms of environmental damage, such as if you have to apply superphosphate to keep your cocksfoot or ryegrass growing, then that comes from Nauru, or it used to. I'm not sure whether it still does. And Nauru's almost down to the waterline from all the mining of the phosphorus that's been laid down over geological time by, or formed out of guano from the seabirds. So yes, farmers understand that from a sustainability point of view and from the point of view of providing pasture that will persist when introduced pastures give up the ghost. Yeah, there's some really serious efforts. Getting seed for sowing in big quantities is the big problem. I think there is stuff like that in that chapter 6 of the notes that I was just talking about, but it'll be fairly rudimentary. And the key reference is that book by Meredith Mitchell called Native Grasses. I don't remember whether those notes mention that book. I'll just scan up and look whether the title and other details are in section 1.2. No, they're not. It's just a small spiral bound book called Native Grasses with a longer subtitle. If you're keen, I can find my book and bring it along tomorrow. Would you like me to do that? Yep, okay. I'll try and remember to do that. So there's the book and there's the plant press. Okay, any other questions or comments arising from last time? No? Okay. There are some issues to deal with. I'm going to share a screen which is on a little camera I've got set up attached to the microscope, because we looked in that wind at the lobed wallaby grass, and I've got a seed of one over here, and it was very hard for people to see these little auricles that distinguish it from any other wallaby grass. And it's even hard to see it here. I'll point out where it is. We're looking edge on at one of them, which is over here, so you can't see that. I can just see one here. I'll try and focus it a little bit better. I think that's maybe slightly better. So I'm looking at this point. I'm going to roll it around a bit so that you might be able to see it better. There's a time delay in this, so that makes it rather hard. I had a Florida for the morning group and it was really obvious. This one's not going to play the game. Okay, now if I can use my left hand backhandedly, I'm looking at a little flap of tissue there, little triangular projection. And if I roll it around, you can just make it out there, I hope. Yeah, where the arrow is pointing. If I roll it around on the other side, there will be the same sort of auricle on the other side. And let's see if I can, yes, I can see it there. Okay. Just have to grab the thing, hold it on the right angle. It's proving very difficult. Okay. Yeah, I'm really having difficulties, but I think I can do it now. Okay, I can see the two little triangular projections there. I can see one sticking out there. If we think about that unicorn, and this being a unicorn grass with the tip of the snout there, and these being the two ears of the unicorn, and here is the horn of the unicorn. On the outer edge of this ear, it has a very thin wing on it, if you like, and the wing is projected up into this little point. And the same on the other side, we can see a wing there with its edge, and then a little projection there. And that's all it is that makes this the lobed wallaby grass that you will find recognized in an identification key as a rule. There are other things, like I said, that I could spot them from a distance. So the plant looks a bit different, the seed head looks a bit different, it's a bit more biased to one side of the stem and all that sort of stuff. But the methodical ways of going about identifying this species really rely on you spotting that. And out in the wind, that was very difficult. So I thought I'd better show you what you should be looking for. Any questions on that before I move on?
Yeah, Graham, what's the term for that little projection?
They're usually called auricles. Aural means to do with your ear, so it's like a couple of little ears. The common name of lobed wallaby grass refers to these as well, so you could call them lobes, but there is a complication, which is that in books and in my key, one normally calls the ears of the unicorn the lobes. So this is a lobe, but then this is like a lobe on a lobe. So you can call them lobes, but it's sort of a second order lobe. It's a lobe on a lobe. And to avoid that confusion, I suspect they're usually called auricles. And the scientific name is Ritidosperma auriculatum, and that refers to these auricles.
Thank you.
Any other questions? Okay, then we'll go on to the next follow-up from last week, which is that I mentioned that there are two species of Themida in Victoria growing naturally, and I told you that the way you can tell the plants apart is not by anything to do with the overall plant, because they're pretty much indistinguishable, other than if you look at the individual seeds. And I have here seeds of the two species, the kangaroo grass, Themida triandra, and the oat kangaroo grass, Themida avanacea. And when the seeds are ripe, this is what will fall off quite commonly as the seed. Or you will often find that some of the seeds don't have these peripheral bits. So the grain lives down in there in kangaroo grass and in there in oat kangaroo grass. And this part is what we would call the fertile spikelet. Fertile because it has a grain that develops in it. There is an awn sticking out the end. Equally on the kangaroo grass, we've got the fertile spikelet and its awn sticking out there. Now, I reckon this looks a lot like a speargrass seed, don't you think? Leave aside these other bits that are attached, but don't you think that looks like a speargrass seed? The thing is, the speargrass seed is not a spikelet, it is called a floret. A spikelet includes glumes. And these surfaces that we're looking at there and there are not not lemmas. They're not, we're not seeing a floret, we're actually seeing glumes. So we've got a hairy gloom on this oat kangaroo grass and a rather shiny, smooth gloom on kangaroo grass. And that's how you tell the two apart. And as I mentioned last time, I'm familiar with this species at Charlton and Boort, and I should have checked because I have a feeling that there's one other site in the river that's in the Riverina where the oat kangaroo grass occurs, as well as in New South Wales and up into Queensland. Queensland has something like 4 species of Themida, including greater grass, which is introduced from overseas and is an annual species. And there are 3 or 4 native Themida species, and they all look like kangaroo grass. So if you, well, I know that Carla and Cam are out in that general part of Victoria. I know Lockwood's still a fair way away, but you may be out in that part of Victoria, and this is how you would tell the two apart. Now, let's talk further about these bits that are attached to our fertile spikelets. It's perhaps a little clearer on the oat kangaroo grass and on the kangaroo grass here. You'll see that there is a little stalk there attached on the bottom of another, well, it's what we'd call a male spikelet. And there's another one there. And it's quite common for the fertile spikelet of any Themida to fall off the plant with these 2 male spikelets attached. So what we've got there, and what you might think of as a seed, is actually 3 whole spikelets. One of them is bisexual and fertile meaning it produces a grain, but it also produces pollen. And these 2 only produce pollen, and they're on these stalks. Down on the kangaroo grass, Themida triandra, the male spikelets are green at this stage. I don't think this would be right, but it fell out of the plant anyway. And there are 2 of those, and again, they're attached to little stalks. And before these seeds fell out of the plant, surrounding this package of 3 spikelets, there were 4 surrounding male spikelets that look just like these ones, except that they don't have stalks. So they would be radiating from just below the tip of the sharp point on each of these seeds. That's what makes Themida flowering parts, their spikelets, so complicated. We've got the spathes, as they're called, and I'll talk further about those leaf-like things that are in amongst the flowering parts of the plant. And they clasp around a cluster of 4 male spikelets that are distributed evenly on all sides around the fertile spikelet. And alongside the fertile spikelet, there are 2 male spikelets that are on stalks, the so-called pedicellate spikelets. So incredibly complicated. It's just as well that kangaroo grass is readily identifiable from its overall appearance in the field. because if you had to identify it by recognizing what all the different spikelets are, it would be hellishly complicated. And that's what most people have to do if they haven't just been shown the thing and can recognize it from its general appearance. Are there any questions or comments on Phemida species?
Yes, Graham.
Yep.
Can they cross-pollinate?
Oh, between the two species?
Yeah.
I don't know. I certainly don't know about Phemidas. Some grasses do hybridize. In fact, you know, in the plant kingdom, I'm not so familiar with the animal kingdom, but certainly in the plant kingdom, most grass species have evolved as a result of hybridization, then back crossing or doubling of chromosomes and stuff like that. That's how we have the diversity of plants that we do. So hybridization amongst plants is not the main form of reproduction, but it is certainly a very important part of reproduction in the whole plant kingdom. Within grasses, it certainly occurs, but it's still only a minority of all the seeds that a grass produces. And so with Phemida, I haven't seen any evidence of any hybridization. I've never seen any intermediates. So it may be that they don't, but it's very different from orchids, where our Australian orchids in particular have all sorts of elaborate techniques to court a particular pollinating insect in order to not get the pollen from a different species. So it doesn't have, grasses don't have that same level of protection against cross-pollination, but it certainly does happen. I certainly do see intermediates that are convincingly hybrids. I have a plant in my collection here, my research collection, whose seed I raised after collecting it from a plant of Richardosperma auriculatum growing at Toolern Vale where we were, and I pointed out Richardosperma erianthum as well, growing along with and alongside auriculatum. And the seed that I collected from auriculatum grew into a plant whose characteristics are all entirely intermediate between auriculatum and erianthum. And it's convincingly a hybrid between the two. It's also sterile. So most cross-pollination between grass species result in sterile plants. They just won't reproduce. So yes, it may be possible that there is some cross-pollination in Phemida. I've never seen any intermediate specimens that raise that hypothesis. And even if there is cross-pollination, that usually doesn't mean that the offspring, the hybrid, is viable. Okay, I think I'd better move on, or we will run out of time, unless there's any important questions that you want to add. No, okay. Now I'm going to go to a PowerPoint slide because I wanted to show you some close-ups of the seeds of the Ostrostypa cetacea. That was about the last specimens that we looked at out at Toolern Vale on Thursday. We saw that the seeds are black, apart from the hairs that cover them. So here is what I would call a seed of Ostrostypa cetacea. You might remember it had a rather slender seed head. The plants were smaller than any of the other spear grasses that we looked at. In fact, as spear grasses in general go, it's one of the smallest plants. And even from a distance, a metre or more away, I could spot that that's what this species was, in part because the inflorescences had this blackishness to them. And the blackishness was because you could actually see bits of, particularly the ends of the lemma. The terminology with a a seed like this is, we've got a sharp point on the callus. This is hardened, so that's called a callus. And the callus is the lowest part of the lemma, which continues up to where the awn joins on. The awn is there, and it's effectively, well, we normally talk about the lemma finishing at that point. And you can see that there are some hairs around the base of the awn. That would be called a coma, as distinct from a corona that you would find on an ocella. Now, it's got this ball bit. So the lemma, we would say, has hairs fairly uniformly scattered over most of the lemma, and then those hairs abruptly stop at what, well, part of the lemma that rapidly contracts into the diameter of the awn. The species that you're most likely to confuse this with, and that wasn't immediately coming to me on Thursday, just as soon as I saw this picture, I thought, of course, it's Ostrostipa gibbosa. Gibbous means bulging on one side, like a pregnant belly or something like that. So a gibbous moon is when the moon is not quite full and it's bulging on the side of the moon that's got the shadow on it. So we've got a bit of a bulge on this side, you can see. Ostrostipa gibbosa has an even bigger bulge, but it doesn't have some of the other feet. Oh, and it's also black. This part would also be black, but it has some features that are different from Ostrostipa cetacea. And here's one of them. If I roll that around a little bit, you'll see that the lemma wraps around there, and it's got two edges on it with a groove down the middle. Now the groove there is not the lemma. The lemma actually stops around there and there. And the groove is between the two edges, or margins, as a botanist would say, the margins of the lemma. And what we're seeing in that groove is actually the palea. We saw some grasses that were at anthesis on Thursday. That is, they were shedding their pollen, I think. Tell me if I'm wrong there. Certainly in the morning we saw some, and grasses mostly flower in the mornings rather than the afternoons. But I think we still saw some in the afternoons, didn't we? Can somebody nod their heads? Can't see enough of you to know. Well, anyway, when pollen is being shed, this groove would open up and the palea would stick out, and there is the palea. It's about as long as the lemma, and it would just stick out as at an angle, typically about 60 degrees, and you'll see the stamens and the stigmas sticking out for a matter of about 2 hours. And in that time, fertilization would occur and the palea will close up and the grain will develop inside this structure. And Ostrostipa cetacea is unique in having a groove along the palea. So when we look at the seed, we actually see the groove, and the margins of the lemma curl inwards and interlock with the edges of the groove. The groove in cross section is a bit like in a jigsaw puzzle. You know how jigsaw puzzle pieces fit together. In cross section, that's what this is like, and the edges of the lemma actually interlock with the groove in the palea, and it's the only species that does that. And in the and for decades, when I was learning about grasses, I just knew in the key that there was this point where the key to spear grasses asks, does the palea have a groove? And I would always have to get the microscope out and dissect out the palea from inside the lemma and see whether it had a groove. But in practice, you don't need to do that because the groove is evident even without doing the dissection, because the lemma margins actually interlock with it. So there's a tip. However, the key that I've given you doesn't require you to do any of that sort of stuff. It's rather more straightforward. And here is a close-up of that area where the Orne joins onto the lemma, and you'll see the dimples, I'd call them. There's a pair of dimples, one on this side and one on the other side, and it's not really well focused here. I'm not really satisfied with this photograph, but these glistening bits are little shark's tooth sort of protuberances sticking up there. It's really rough. And what sort of evolutionary selection ended up with this, I have no idea, but I find it quite fascinating. There's some secondary dimples over here, so you end up with what look like 4 ridges and interspersed with 4 dimples. And that's enough on Ostrostipes cetacea, I think. Has anybody got any questions or comments?
I've got one, Graham.
Hmm?
Would you say you need a microscope for most Ostrostipes species identification, or is a hand lens adequate?
Hand lens is adequate with the key that I've given you. Because the Vicflora key, well, if you had any speargrass that has a coma, and the coma refers to these hairs that are attached around the top of the lemma, surrounding the base of the Orne, that's a coma. Any species with a coma in the key, I think that maybe it's the first couplet, asks you to inspect the palea. Now, I've shown you that you don't always need a microscope. You can usually see that groove even, well, if not with a, well, sometimes I see it with the naked eye, but a hand lens would certainly be enough on most occasions. But if you don't see it, then it could be that it is an Ostrostipes cetacea, or it could be that it's Ostrostipes cetacea and it just isn't mature enough, or for some reason you're just not seeing it very well. And then you're going to have to dissect it, and that does need a microscope. But the key that I've written avoids that. So I think, George, you'd be able to get by with a hand lens without much trouble, but every home should have a stereo dissecting microscope. They're wonderful things, and you can get a quite reasonable one for a couple of $100. And whereas these photographs that I've been showing you are not all that good in clarity and so on, a dissecting microscope gives you that amazing 3-dimensionality to it with your stereo vision, and it makes things vastly clearer, even although the magnification might be 10 times the same as your typical hand lens. You will see things vastly more clearly with a dissecting microscope. So, Georgie, you work in an office where I'd hope that there would be a dissecting microscope, is there?
Yeah.
Yeah. So I'd encourage you to do that, but out in the field, I know you don't want to have to keep collecting specimens and taking them back to the office, but I reckon you can do it without a microscope. It's just that it does make it easier.
Yeah. All right. Thanks. I'll have your key handy.
Just with the key, Graham, is it meant to have all the species on it, or do we then use the species that it's got on it to then decipher about the other species?
Okay, if you have a look at, well, I'm going to show you this later. Look, perhaps we'll deal with that mainly later, but let me tell you that couplet 30, if you know what a couplet is, look for 30 in the left-hand margin. It will tell you that at this point, I haven't completed that group of spear grasses, and I can't remember how many there are. I'm thinking there's probably about 8 species that I have not yet done. And instead, I refer you to the Vicflora key for the time being, until I send you a link with the completed key, which I hope to do over the summer months. Okay, is that enough for the moment?
Yeah, thank you.
Okay.
I
Let
have a
me now
question.
move on to the complexities that can arise in grass species that have evolved deviations from the more simpler kinds of grasses, the more ancient ones. Here's a case of a typical simple sort of grass whose spikelets contain a pair of glumes, which in most grasses remain on the plant once the seeds are shed. And inside that pair of glooms, there may be just one seed, and that would be for the whole tribe within the grass family, or there may be multiple ones. And in a tribe that has multiple seeds in each pair of glooms, all of the species do. Equally, a tribe that has one species that has only one seed in each pair of glooms, and all of them do. So we can split the grass family more or less down the middle between those tribes that have only one seed within each pair of glooms and those that have never only one. In this diagram, which I've modelled on a POA, we can see that there are, what, 1, 2, 3, 4, 5 seeds that would be shed at maturity. And each one of these seeds we would call a floret, with a grain developing down in each one of these. And each of these seeds or florets comprises a lemma, which is the larger, more visible part, and a palea, which would be partly or wholly hidden by the lemma, as in the case of the spear grasses that we were just discussing. And there would be an ovary and some stamens in there to allow each one of these things to develop a grain. And at maturity, the little stalk that runs up the middle, the racilla as it's called, would split between these. The top one would split first and shed, and so the first floret with its lemma and palea and grain would fall out first, and then the racilla would split there and there. Disarticulate is the botanical term for this splitting process. And finally, the last one of these down the bottom here will disarticulate, leaving just the glooms behind. That would be the simplest, most ancestral formula for a grass plant. But there are variations. And let me give you the example of Ehrhata longiflora, the annual veldtgrass, which is one of the species that I handed out specimens of out at Toulon Vale. So and I've ended up with some leftovers, so I'm sure a couple of you might have missed out on getting the specimens at all, but most of you should have a specimen of annual veldtgrass. And what you're looking for is something that has seeds like this and branched stems. I'm going to show you those branched stems. I'm going to stop sharing that, and I'm going to swap to the camera at the rear of this computer and get my specimen. Now here you can see that I've got a stem with some roots on the right there, and if I go up a little bit further, I can see that the stem is branched. And if I go up a little further, I can see that within this leaf sheath that I'm folding down, I've got the oldest of the stems, the thickest one. I've got a second one there, and I've got a third one as well. So it's got two side branches arising from the node of this leaf. And if I go further up, I can see further branches. And what happens with annual veldtgrass and a whole range of grass species is that once they've flowered at the end of the stem, well, firstly, they may start to flower at the end of a stem before any side branches are produced. And once the flowering gets underway, or in some species, once the seeds are approaching ripeness or have even shed all their seeds, then the grass will start producing side branches with seed heads or inflorescences at the end of those. It's a particularly good year for annual species of plants, including the annual veldtgrass, and so there's a lot of branching going on. A young plant, you won't see that branching. So that is, oh, there was one other thing that I was meaning to show you, and that is the leaf sheaths. You remember I pointed out that some grass species have got sheaths that are folded along their spine, so to speak. So if you were to grasp your specimen around a sheath and roll it around in your fingers, you can readily feel, not just see, that the sheath is not round because it's got this fold along the spine. And that's true of Earhata erecta and Earhata longiflora, this specimen, and to a lesser degree, Earhata calycina, the perennial veld grass. And that's a very useful identifying feature for those of you who want to identify grasses when they're not in flower. The other thing that goes along with Earhata erecta and Earhata calycina and Earhata longiflora is that when they're seedlings, or very young shoots, they have a characteristic reddish purple coloration to the outer sheaths. So even as the seed first germinates, you can spot it, at least as one of these Earhatas, by the reddish purplishness and the fact that the sheath of even that first leaf has the fold along its spine. If you then pull up your plant gently, you will see the actual seed attached to it, and it will be identifiable just from the seed, even if it wasn't for those other things that I've just told you about. And I'm just checking the chat, and I see Maddie's asking, hey team, is there a link for these slides? No, there isn't. Now that's a good point. I should make them available, shouldn't I?
That's all good. I think Sam said the info's in the handout, so that's all good.
Okay.
Thanks.
Yeah, the information is in the main lot of notes, even if the slides aren't. Okay. Now, I need to go back to my PowerPoint slides. What you've got with your annual veld grass is a plant whose seeds look like this, with a pair of husks, a pair of awns attached to each seed. It turns out that hardly any grass species in Victoria have just the two awns or bristles on each seed. Weeping grass is the one that looks closest to this, and weeping grass was originally described as Earhata stipoides. In fact, in New South Wales, they still tend to call it Earhata stipoides rather than Microlina stipoides. I'll show you that species tomorrow, and you'll see that there are loads of differences between a Microlina and an Earhata. So there should be no confusion between this species and weeping grass or Microlina stipoides, but they do both have two awns on each seed. The other species, of which there are only about 3 that I can think of. Oh, well, there's 2 vernal grasses, annual vernal grass and sweet vernal grass. They've both got two awns on each seed, and they don't look remotely similar from the scale of the plant to the details of the seed. Nothing would allow you to confuse a vernal grass with an Earhata or a weeping grass. And the other ones equally couldn't ever be confused with annual veld grass. So it's easy to identify. Let's have a look at what the seed comprises. Hopefully your seeds are looking pretty much like this. You'll notice that there's a little tuft of hairs at the bottom, like a little tickler. And then we've got an interesting looking ridge there on this outermost husk. And then joined into the base of that husk, there is a fascinating looking hinge here on the second husk. And if it were a floret, that's the limit for the number of husks, because what we call a floret in grasses comprises a lemma and a palea, and the ovary and the stamens inside. So only ever a maximum of 2 husks, the lemma and the palea. Well, in this case, we've got 1 husk there, 1 husk there. Then we've got a third one. And if we follow that edge down, it goes down there. And then we've got another husk in there. So it's actually got 4 husks. How do we interpret that? The interpretation is this. We can call it all one seed, but it's actually got 3 florets. And 2 of them have evolved away their ancestors' role of producing a grain. This is what we call a neuter floret. Neuter because it doesn't produce any pollen and it doesn't have an ovary that develops into a grain. So is this one. The ancestors of the Earhartas and Microlenas would have had probably 3 florets, and all 3 of them would have produced a grain. But in the Earhartas and the Microlenas, the 2 outermost ones, or the lowest ones, if you like, no longer produce grains and instead have a completely different function. They protect the grain-bearing floret in the middle, and they also aid dispersal because these are very raspy if you feel them. And as these wriggle around on the ground, and they don't have a geniculate horn, so they're not actively wriggling around with moisture, but they do have horns that are very raspy, and that helps to make them wedge into corners and that sort of thing. And right in the middle is the fertile floret, the one that actually develops a grain. And this is not too uncommon in the grass family, particularly when you go into the tropics. So the principle involved here is that some of the more highly evolved grasses have sacrificed some of the seeds that their ancestors would have produced and instead use these modified forms of florets to protect the grain-bearing parts in the middle of the seeds and often to aid dispersal. In fact, they pretty much always aid dispersal, but they also have a protective function. So that's what we're seeing here with this Earharta longiflora. Same sort of thing with the panic veld grass, Earharta erecta on the left here. And there's the glumes. Falling out of the glumes is something like that, which is then on the left on this illustration here. And we have the same structure of hinge, fascinating sort of thing. We've got a lower neuter floret there, an upper one there, and clasped in between these 2 firm textured florets is the grain-bearing or fertile floret in the middle. This occurs in the veld grasses, Microlena, all of the panicoid grasses such as panicums and Bolwellia. It occurs in the vernal grasses and in Yorkshire fog and a range of other grass species that aren't all very closely related. So it does happen a fair bit. Now, most keys to identify grasses will rely on the person using the key getting the seeds and realizing that the seeds contain sterile or neuter or sometimes male florets as well as fertile ones, and that can be really difficult. So I worked out, I worked on the key so that you do not need to sort out which are the neuter bits, which are the male bits, which are the bisexual or fertile bits. Instead, I've used other characters, as you'll see later on in this session. But if you're using books, you will see frequent reference to sterile florets, and a sterile floret can be either neuter or male, depending on which book you're reading. I think it's crazy to call something male neuter. I'm male, but I'm certainly not neuter. I've got a son. But you will find that in the literature about grasses. They'll use the term neuter or sterile. Sterile can mean male or neuter. I'm using the term neuter to be very specific in this case. So that's just by way of warning that there are complexities that most books will expect you to already understand. Now, we're going to use some keys. And the first key I want, perhaps I should go back. Has anybody got any questions on that, or those variations that have resulted from evolution? No, I hope people aren't falling asleep. Keys won't make you fall asleep either. Let's try identifying a wallaby grass. I'd like you to get your specimen that I gave you, except for those of you who never got any specimens, get your specimens. There are 2 wallaby grasses in the set. One of them has a much more elongated, slender seed head or inflorescence than the other, and that's the one that I want you to get. And also, you might want to get out your key to wallaby grasses either on a screen or as a paper copy, because we are going to identify that wallaby grass that has the more slender seed head. Now, by way of introduction, I just thought while I have my PowerPoint slides up, I will highlight one of the main differences between wallaby grasses that are used in identification keys, and that is the arrangement of hairs over the lemma, or what we might think of as a unicorn's head. Now, Kate, you missed out on last week's session. In that session, I likened the seed of a wallaby grass to a unicorn's head, where if I take this one at the bottom left, which is quite distinctively the brownback wallaby grass, which was in the drain on the side of Missons Road last Thursday, I would liken it to having the tip of the snout of the unicorn there, and here is one ear here, here's the other ear bending over a little bit, and here is the unicorn's head. But unlike most unicorns, this unicorn has got a bend in its horn. So that's the analogy with the unicorn, and I'm going to use that analogy to be able to communicate with you about the different parts of the unicorn's head, if you like, but what is technically known as the lemma. So I've oriented each of these wallaby grass seeds so that the lemma is facing us, the unicorn's face, if you like. But we can also see the palea in between these 2 ears of a unicorn here. I can see sticking up from the back the top of the palea, and it's rather transparent there. And I just remembered I meant to point out the palea on the lobed wallaby grass too. Here's another species. This is not the brownback wallaby grass. I won't even tell you what it is. This form doesn't occur in Victoria. The species does. And you can see the palea sticking up there. In both of these species, the hairs are seen at 3 different levels of the unicorn's face, so to speak. We've got some at the very tip of the snout, as you see on every species, and that's true of every species of Ritidosperma. There is always a pair of tufts, one on each side. I think you can probably see that they're on each side on this species most clearly, because it's got a broad callus there that isn't so obscured by the hairs. All wallaby grasses have a pair of tufts, or sometimes lines of hairs along the callus, as it's called. Most species of wallaby grass have hairs displaced a little bit above the tip of the snout, typically 1 or 2 millimeters. So you can see a rather patchy row of hairs on the brown-back wallaby grass. I see a continuous row of hairs on this species. And there, it doesn't look quite continuous, but it does have a row. This one has a row of hairs too. This one seems to have hairs scattered all over the unicorn's face. On this species, at around about the unicorn's forehead level, you have a continuous row. Well, I say continuous, and this is an important point. You'll see that it's actually made up of a line of tufts of hairs. And you'll also see some ribs running along it. And here you can see some lines running along it. Every wallaby grass has got 9 veins or nerves, the ribs if you like, running up it. And the hairs on a wallaby grass seed are always in tufts in between those ribs. And there are 9 ribs. So that means that there are 10 tufts, because there's a couple between the edges and the outermost of the nerves or veins. So we've got a full complement of 10 tufts of hairs on this brown-back wallaby grass. This one seems to be missing at least some of that tuft there, but it's pretty much a continuous row of hairs. This one, well, it's got a continuous row of hairs and they're quite long there, but it's also got hairs that are a bit shorter scattered all over the face of the unicorn. This one's got a row of very long hairs, and so does this one. This one doesn't seem to have any hairs scattered between those 2 rows, and this one does have a few scattered ones there. So these sort of different arrangements of hairs, and whether there's the full complement of 10, what we might call a complete or continuous row of hairs, versus some like the ones that I've given you the specimens of, where that row is missing. They're the sorts of things that a lot of store is put upon when you go to identify a wallaby grass. Any questions before I leave that slide and go to the key to wallaby grasses? No? Okay. Then I will do just that. Okay, hopefully everybody's got some notes that look like this. And there's some introductory notes there talking about all the different names that have been given to different wallaby grass species over the last probably 15 years. Well, it's probably 20 years since we've called them Danthonias. I will show you a Danthonia tomorrow, a fair dinkum Danthonia. We never used to call it a Danthonia, but now it's the only Danthonia species left in Victoria. But it's not a wallaby grass. Okay, there's some information about where Retrodospermas occur around the world. They're not just in Australia. And some important things. I've told you about the Palea. I've talked about the unicorn analogy, which we'll find quite useful. I've mentioned the callus and the Palea. The length of the seeds is very important. And in order to measure it, 2 things. Firstly, if you have a look at your specimen, what I'd like you to do is pull some seeds out from inside one of your pairs of glooms and see how many there are. Now, it may have already shed some because I got material that is ripe, but try and pull them out as carefully as you can so that you get the full number and try counting them. You may find that if you've got one of the less ripe spikelets, the florets or seeds are sticking together, and that can be quite confusing. And for that, whilst you're still holding that bunch of seeds or florets, you can give it a push with a finger on the pointy end, and that will break it up into the individual seeds. I'd like you to do that. You'll find that, well, clearly there's more than one seed. How many seeds are you getting from each pair of glooms? I'll have an auction here.
I've got 4.
3.
4 or 3. Gee, I reckon it's lost some. Try another spikelet. Do I hear an advance on 4?
I've got 8.
8, good. Right, now you're really, I reckon you might win with 8. Anybody got, how many?
8 as well.
8 as well, good.
Yep.
Occasionally you'll find you might get 9 in this species. Not often will you get more than that. And I'm thinking that at least those of you who found 8 will have noticed that the size of the seeds or florets becomes smaller the further you go from the glooms. And that's normal within grasses in general. If there's more than one seed in each pair of glooms, the biggest ones will be down next to the glooms, and they will get progressively smaller as you get further away from the glooms. If you're going to measure a seed, the key will always assume that you have a fully developed one, not one of the runts that are furthest from the glooms. So you always try to get one of the biggest ones. It also makes it easier to see them because they're not so tiny, and some of the bits that we're looking for, such as those little tufts of hairs that make up the rows, they're hard enough to see even when you get the biggest of them. You don't want to make it even harder by getting an unnecessarily small one. So those of you who said that you found 3 or 4, did you find more in another spikelet?
Yeah.
Yeah. Up to
Okay,
6.
good.
I haven't found them with 8 yet.
Okay, good. All right. Now, the official way to measure the length of a wallaby grass seed, other than in Flora of Australia, if you happen to have volume 43B, I think it is, of Flora of Australia, you won't read this unless you happen to read the fine print at the start of the whole section of the whole subfamily. You might just go straight to wallaby grasses, and it won't tell you there, but I can tell you that the key in Flora of Australia says to measure from where the awn joins on in between the two lobes, as we would technically call them, between the two ears of the unicorn. Flora of Australia measures from there down to the top of the callus. And by the top, I don't mean the tip, I mean the bit that's, well, you're measuring from where I'm gesticulating here, the top of the callus, up to there. But that's the only reference I know of that measures them that way. And because of that, people who've used the Flora of Australia key have ended up with countless misidentifications because they've got a measurement which is a millimetre or two too small. And the correct way of measuring is from the tip of the snout of the unicorn to the point where the horn joins on between the two ears. That is the way you measure the length of a lemma, the lemma being the face of the unicorn. Okay, I think that's all I need. Oh no, there's one other reference there at the bottom of the page. And in this illustration on the notes, you can see that the twisted part, well, it's not twisted, it's coiled. It's like curling ribbon, which makes it different from all other grasses in Victoria with these geniculate awns. And Kate, a geniculate awn is an awn that twists or coils in the lower part and develops a bend. Geniculate means bent. And this happens as it dries, and then it uncoils when it gets wet, such as if you put a drop of water on it. So Kate, you didn't see the neat little party trick last week. I suggest you put a drop of water on one of these coiled parts of the awn on a wallaby grass after we've finished, and you'll see it wriggle around and straighten up within seconds. Oats are probably the best one for doing that, except that they don't jump. I like the sweet vernal grass with jumping seeds. So try any of those. Okay. The key doesn't include Ritidosperma australe, the Austral sheep's grass. It's not a wallaby grass. You wouldn't confuse it with one, but it is put in the same genus now. And I fear that it may have even become extinct in Victoria. It occurs naturally in on the Kosciuszko, on the highest parts of the Kosciuszko Plateau, and in Tasmania, in the Alps in Tasmania. But I think it's probably become extinct in Victoria because I just can't find it. It was always rare, but it seems to have become extinct in Victoria. And it's not in this key. Okay, now, many of you have used a key before. Those of you who've used a key to grasses will have found it extremely frustrating. Let's see how we go with this. We're going to identify the specimens that you now have. Or in case of Kate and maybe one or two others of you, you'll just have to rely on the images. And I'll tell you that the species that we've got has seeds that look very much like on the front cover of this lot of notes. So if you don't have the specimen, as we talk about the seeds, you can use that illustration and that will do. Okay, now there are two different sorts of identification keys that are used for plants and animals, more for plants than for animals. There's a kind that's been developed over the past 15 to 20 years, which you can call an interactive key. The brand name of the main kind is Lucid, developed in Australia. A lucid or interactive key behaves in a different way. It has a database with all of the different ranges of characters of a grass. For example, the length of the awn and whether it's perennial or annual, how wide the leaf blades are. It's got a database behind it with all of those sorts of things. And the interactive or lucid key allows you to choose any character you want, such as how long the awn is. And you type that in and the software will sift out to leave you with a short list of all of the species of grass within Victoria, if you've got a Victorian interactive key, all the Victorian species that can produce awns of the length that you've specified. And then you can look at your plant and say, what else is going to be good for me to specify? I'll specify how many seeds are inside each pair of glooms, and you type that in as maybe 2. And that will narrow, or the software will narrow it down. It'll interrogate the database, query it as they say, and come up with a short list that's even shorter than before. And progressively, you can narrow down the identity of the plant just by eliminating the things that couldn't possibly match the features or characters that you're specifying. And the software will also give you hints as to which sorts of characters are going to give you the greatest amount of discrimination and split your shortlist down into a smaller category. So they're good, but they tend to have quite a lot of bugs in them. The databases don't have enough data, and so they end up giving you wrong information. The lucid key for grasses was called Ozgrass, and it was produced by the CSIRO, and the CSIRO hasn't paid to upgrade it to the current version of Lucid, and so people like me who bought the thing can't use it anymore because it's not compatible with modern software. So there are interactive keys, and one will eventually be made available, I expect, on the VicFlora website, which already has interactive keys for Asteraceae and is it Fabaceae? Who knows what the other one is that was released recently? Anyway, you'll find a couple there, but not grasses yet. The alternative, the more old-fashioned way of doing it, is called a dichotomous key. A dichotomy, of course, is where you're faced with two alternatives you have to choose from. And that describes the way the key works, because in a dichotomous key like this one, down the left-hand margin, you'll find things in pairs. In this case, there's a 1A and a 1B. In, I think Flora of Australia has got a 1 and a 1 colon and that sort of thing. But you'll find things in pairs, forming a dichotomy, or what in keys are usually referred to as couplets. So 1A and 1B form a couplet, and 1A is the first lead of the couplet, and 1B is the second lead. It's called a lead because it leads on at the end to the number of another couplet, and it contains text which, well, one and only one of the two leads in a couplet should be a perfect match for your specimen. So we've got a description of the grass in 1A and a description in 1B, and our specimens should be a perfect match for one and only one of those. If it's 1A, then we'd go to couplet 2, otherwise we'd go to couplet 8, and you work through these dichotomies or couplets until ultimately you get to a point like 2A here gives you that instead of going to another couplet, you've got Mutadisperma nivicola in that case. Now I see there's something in chat. Eucalyptus, thank you for that, Rose. Yeah, okay, I'd forgotten. Anyway, if you go to the VicFlora website on the home page for VicFlora, it will tell you the 2 or 3 interactive keys that are available. Okay, let's see how it goes. I'll read 1A. It begins, above the middle of the lemma body, most hairs are in 2 tufts, one near each edge of the lemma, perhaps with sparser, shorter hairs in discrete tufts along a line between the 2 side tufts. In other words, instead of having the full row of 10 tufts, we are looking at the situation where there are tufts at the margins, the edges, and a whole lot of missing ones, maybe with a few hairs on the midline of the lemma or the unicorn's face. Now, I'm going to spend a bit of time on this because the greatest problem that people have with using any key to wallaby grasses, including this one, is this couplet, where the hairs are and whether there are hairs scattered all over the unicorn's face and that sort of thing, because people are, by and large, not used to having to look at things up really close, particularly really tiny things. And people take a first look at a floret of a grass, of a wallaby grass seed, and they think, oh yeah, I don't see hairs there. Or they think, oh yeah, I can see that this sort of rose there, it'll be this. But they don't look closely enough. You really do need to look very closely. This is where a dissecting microscope is really good. And prior to the pandemic, when all of my classes were done in person, I had microscopes so that everybody could see these things under the microscope and could develop a good mental picture of what these things look like. You don't have that benefit, so to say. So you're going to have to place more emphasis on how fastidious you are at looking where the hairs are attached onto the lemma or the face of the unicorn. I'm going to stop sharing my screen just so that I can show everybody what it looks like under a microscope. Where's my microscope? That's funny. It seems to have gone. Oh, there it is. Okay. Let's get rid of the stuff that I was showing you before, and I'll put on a seed from one of the same specimens as I gave you. Incidentally, I'm putting this on the sticky side of a Post-it note. Now, it's not strictly a Post-it note, but it's the same sort of thing with the sticky stuff, because that stops them wriggling around. Now, I want to orient it the right way. Peg it down there, move it into position. And get it focused. Okay, that is what I am looking at. And we're talking about above the middle of the lemma. So I've got to silence my phone again, excuse me, I'll just do that. It says it already is. Goodness knows why it is still making noises. Okay, there's the lemma extends from the tip of the callus up to where the horn joins on. Oh, be quiet, phone. There's the middle. So above the middle, I've got some hairs along the line there. And it's a bit hard to see quite what they look like. I'm going to have to move it a bit because I'm going to zoom in. And if I zoom in, it's not going to be in the middle of, or in the field of vision. So I'm going to put that right in the middle. Then zoom in. And refocus. Right, now you can see the tip of the snout there. The higher you zoom in with a microscope, the less depth of field you've got. So I can't get it all in focus, but there's the callus down the bottom here. If I can get that in place, and you can see the tuft of hairs on each side. Then a little further up, we've got the row of hairs that you might liken to being on the moustache of the unicorn. And then we've got just above the middle, this lot of hairs. There's some at the margins, and there's another lot the other side, which are not clearly visible. And we've got some attached there, but there's this expanse, which is a gap in the row. Doesn't matter how well I get it in focus. I think that's in perfect focus there. You can see that there is a tuft there, and if I had a better microscope, you would see that that's in a tuft, and there's another little tuft the other side of the central rib or nerve or vein, and that's all you get. If you're having trouble seeing that, you wouldn't be alone. A large proportion of people struggle to see these things until they get practice in looking at them. And one of the problems that people have is that on a wallaby grass, these hairs that you might think of as being like a moustache of hairs stick up, covering the surface or obscuring the surface of the lemma. And so people don't notice what's going on behind the screen formed by the moustache of hairs. And so they're not careful enough about whether there are hairs actually attached on this part. And so what you will often have to do is push those hairs down. And there is a time delay in what I'm doing. You have to do that sort of thing. The time delay makes it very hard to demonstrate that. Now it's gone out of focus. Anyway, that's the sort of thing that you often have to do. So I will stop sharing that. Any questions so far? No? Okay.
Just confirming, Graham, we're only meant to be looking at the unicorn's front face, not the back of its head.
Correct. It's the lemma, not the palea. Around the back of the head, you will see that more transparent, less interesting husk called the palea. There are parts in this key where you will be asked to look at the palea, but not in this case. Okay, so 1A said above the middle of the lemma body, most hairs are in two tufts, one near each edge of the lemma. I'm going to give that a tick. Perhaps, well, in our case, it does have sparser, shorter hairs in discrete tufts along that line between the two side tufts. In fact, there were just two little tufts. Is that consistent with what everybody else is seeing? I hope it is. Okay, I'm seeing some nodding. Okay. Right, so far so good, but every part of a couplet must match. If there's anything wrong, it's not your species or you're in the wrong, going the wrong direction. Okay, it goes on. The photo on the previous page is a typical example, and I said that I reckon that photograph on the previous page is quite similar to what we're looking at. And there's a tip there that you may need to rotate a lemma or a floret around side on, particularly when you're out in the field, and particularly if you've got some good light, like sunlight on your floret. Rotating it allows you to see the hairs more clearly than if you just look at it face on. So that's a tip particularly useful for in the field. Now, we look at the alternative 1B. It says there is either an uninterrupted row of hairs across the unicorn's forehead, and by uninterrupted, it means the full 10 tufts. It doesn't mean that there aren't tufts. It means that all 10 of them are present. So unless you had a microscope or you're doing well with your hand lens, you will just see it as a continuous row. Okay, so there is either an uninterrupted row of hairs across the unicorn's forehead, or hairs scattered over most of the lemma body, or, as in this illustration, both. And I'm going to say, no, we've got a 1A job, and after the ellipses, it takes us to couplet 2. Any questions before we go on to 2? I'll give you a tip at this point. It can be useful to keep what's often called a breadcrumb trail based on Hansel and Gretel. You remember Hansel and Gretel, as they went through the woods, would leave some breadcrumbs so that they could find their way back. Now, it didn't work for them, as I recall, because the birds ate their breadcrumbs, but it can be useful in this case. Your breadcrumb trail is not going to disappear if you write it down with pen. The way it works is I would, at this point, be writing 1A, 2 on a piece of paper to show where I've got to. And in a minute, I'll put either an A next to the 2 or a B next to the 2. And the reason for doing this is that if you get to, say, couplet 25 and you reach a dead end and you then think, oh, I was a bit worried about that couplet further back. Where was that? You can't retrace your steps. You had to have taken note of it at the time. There's no way of reversing through the process. You can only go forward unless you've kept a breadcrumb trail. The most common reason why people have to go back is that they have correctly identified which of the 2 leads in a couplet applies to their specimen, and they nevertheless choose the wrong thing in the right-hand margin and go to the wrong couplet. I've certainly done it many times myself. And if you go backwards through your breadcrumb trail, you'll see that. You'll think, oh, how did I do that again? So that's something to be wary of. Okay, 2A says the largest florets are 3 to 5.5 millimeters long, including the awns. Has anybody measured their floret, including the awns? What are you getting?
Longer.
How much longer?
Orn's about 12 or 13, and yeah, okay. I don't
Vastly
know.
longer, right?
Yeah.
They're very much longer than 5.5 millimeters long when you include the awns. So, well, we could stop there because there's something seriously wrong, but we should check the other bit. The other bit says a small alpine grass of bogs and wet heathland. These specimens came from Boronia. So it misses out on both scores. It's not Ritidosperma nivicola, the snow wallaby grass, which has by far the smallest seeds of any wallaby grass, but not compared with the Ritidosperma australe that I said was austral sheep's grass, or southern sheep grass. Yeah, southern sheep grass. It also has really tiny seeds. 2B says the largest florets exceed 6 millimeters long, and indeed we've got around 12. That's one measurement we've got, including the awns. Various habits go to 3. So on my breadcrumb trail, I'd be adding to the 2 or 1. I had 1A, 2. I'd now put B, 3, and start with 3A. 3A says the stem has no nodes above ground level, even when mature. We've got mature specimens. Have a look at your stem and you will find nodes. Some of them will be readily visible. Some of them will be hidden inside leaf sheaths. So you might need to peel a leaf sheath away in order to see the nodes. But I reckon even without doing that, you'll see at least one node. I have no trouble doing that on my specimen. Of course, each node has a leaf attached to it. So in principle, you could just count the number of leaves on your stem. But there is a catch with that. Leaves can get chewed off or mown or get pulled off when I collected your specimen. that sort of thing. So the fact that I can see several leaves up my stem does mean that I've got nodes there. But if it really becomes important to know the number of nodes, it's usually best to count the actual nodes rather than just count the number of leaves. Okay, so I'm saying that grasses are more like 3B, which says stems have nodes above ground level by the time flowering begins. So 3A won't work, but we could look at the other bits. 3A goes on, lemmas are hairless except for the tip of the callus, the unicorn snout, and 1 or 2 side tufts on each side of the lemma. In other words, it doesn't have a complete moustache, if you like, and ours does. And then it says alpine or subalpine. Again, we'd eliminated on that basis. So 3A is no good. Next to my breadcrumb trail where I finished with 3, I'd put B, 4 and go to couplet 4. Yell out now if you've got any questions or comments. Okay, 4A says full-size lemmas exceed 3 millimeters long. Have you measured any? Remembering that measurement is taken from the tip of the unicorn snout to the point where the awn joins on. This species is pretty easy to do as far as they go. I'm waiting for a vote.
About 5 mils.
5 sounds about right. If you've got something that is 3 millimeters or less, you've got one of the runts from a long way away from the glooms. So I'm going to accept that vote for a 5 millimeter thing. That would mean that we would go via 4A to 5 and put that on the breadcrumb trail and read 5A. 5A says the tip of the palea extends beyond the base of the central awn by typically 1 millimeter or more, and always by at least quarter of the length of the darker part of the central awn. Have a crack at measuring that. It's getting a bit fiddly. Microscope is really good. I'm going to put my microscope on the screen so that you can see it. And a ruler. I'm getting tangled in knots here with wires and everything, but I think I can do this. I've got to get the focus back. Okay, they are 1 millimeter gradations on the ruler on the left. And we're measuring from the base of that awn where the cursor is up to the top of the palea. Now I can't quite see the very top, but it goes at least that far up, I think you would agree. So with that, that's more than a millimeter, comparing it with 1 millimeter gradations on the left. So, yep, it's more than a millimeter. And then the comparison was whether that length, a bit more than a millimeter, is more than quarter of the length of the twist or coiled part of the awn. So I'll get that in better focus. There's my coiled part of the awn. If I was in any doubt, I would put a droplet of water on that coiling part and it would straighten out within seconds. But even allowing for it elongating by 10 to 25%, I've calculated that and verified it in my measurements. It's still more than quarter, I hope you'd agree. Everybody happy with that? No contest? Good. Then I'll stop sharing that screen and go back to the key. Where were we? Okay, the tip of the palea extends beyond the base of the central awn by typically 1 millimeter or more, give that a tick, and always by at least quarter of the length of the darker part of the central awn. I'll give that a tick too. And it says there in marginal cases to put that drop of water on. Then it goes on. At maturity, the colour of the darker coiling part of the awn is typically pale brown to mid-brown, and the inflorescence is quite slender for a wallaby grass. Well, you've seen a few species of wallaby grass now, and I'd say this is the most slender of all the ones we've seen. It's not the most slender of all of them, it's the second most slender, but it is pretty slender. Okay. Now 5B on the other hand says either the palea does not extend beyond the base of the central awn, or else it exceeds it by less than 1 millimetre and less than quarter of the length of the dark part of the awn. No, that can't be right. Also, at maturity, the colour of the dark coiling part of the awn is mid-brown to almost black. Ours I'd call mid-brown. It's a bit subjective, so that might be okay. But the fact that the earlier part isn't okay means that the whole thing is no good. And finally, it says the inflorescence is not particularly slender for a wallaby grass. And I think this is, although that's subjective. On balance, I'd have to go with 5A down to 6. Do you reckon you'd be able to get this far? Yeah, I'm seeing Maddie nodding there. That's good.
Just with the description of slender, how would you, as you say, it's each to their own, I suppose, but what would you say is slender versus not? Like, I guess when the other day we saw some wallaby grasses with quite round, equally sort of square on either side inflorescence. So would that be less slender than this one?
Yeah, I'd say so. As by slender, I'm thinking it's the width of something versus its length. So if it's really tiny, it might not be slender if the width is still comparable with its length. And so it is subjective, and you get used to it once you've seen a range of wallaby grass species. But this is, I think, the second most slender of all of the non-alpine ones. There are a couple of those alpine ones that were higher up, Nuvicola and Nudiflora, are more slender. There are some that are incredibly broad. In fact, I might bring my Richardosperma vicariae tomorrow. And you'll be blown away by that one. Okay, any other questions? Then on the breadcrumb trail, I'm putting down 6 and I'm saying, oh, that's the end of the line for this route through the key, because it's got to be either Richardosperma racemosum or Richardosperma peniculatum. I'll show you both of these tomorrow. So we've got one of those, and you could look at the pictures, and I'm thinking ours looks more like this picture than that one, but we better read the essence of it because these are quite difficult to tell apart in many cases. 6A, mature inflorescences either do not droop or only somewhat. That's a bit subjective, isn't it? Particularly on the, pardon me, on the margins, that can be tricky. The next thing is the hairs at the tip of the unicorn's snout don't, or barely, reach the row of hairs above. And I'm hoping you'll see that the hairs fit that category. Pardon me. My voice is going. Then it goes on about the row of hairs above the tip of the snout. It's dense. See the photo on page 1, which is of this species. And I'd say, yes, I could see when I pointed it out under the microscope that all 10 tufts of hairs are present, and it's quite a dense lot of hairs in that row. So I'll give it a tick. Spikelets each contain at least 6 florets. Well, we were getting 8 on some of them, and I think everybody ultimately found at least 6. And one of them might be tiny and not readily discerned unless you're really looking for it. So I'm going to give that a tick as well. Then in parentheses, I've said to the trained eye, the unicorn snout is more slender than other wallaby grasses, and the upper part of each lobe contracts more abruptly into the terminating bristle than any similar Richardosperma. So they're things that you get your eye in for over a period of time. And you'll probably get used to how slender that callus, as it's called, is, the tip of the snout, by finding what's in your shoelaces and socks over the next 2 or 3 weeks, because Ritidosperma racemosum accounts for the majority of wallaby grass seeds that get stuck in people's socks. They are incredibly effective at poking those slender points in between the knitting in your socks or in your shoelaces. Now, if you look at 6b, mature inflorescences normally droop markedly, and I'll show you that tomorrow in the field, but not necessarily in harsh conditions, including in subalpine and alpine environments. Remember, these came from Boronia. The hairs at the tip of the unicorn snout reach or overlap the row of hairs above. Yeah, okay, maybe, which is typically sparse and has gaps in it. Well, I didn't see those gaps, and you'll see in my drawings here. Now, the drawings are not perfect, but I've tried to draw it so that you can see that there's some gaps in that row of hairs, the moustache row, if you like to think of it that way, as compared with this continuous row of hairs made up of all 10 tufts on Ritidosperma racemosum. And finally, spikelets each contain up to 6 florets. So if you only found 6, that would be consistent with either of these species. Some people were finding 8. That would only be consistent with racemosum. We have Ritidosperma racemosum, or clustered wallaby grass, or one of my past students decided should be called nature strip grass, because it's just so common that around Melbourne you can see it in numerous nature strips. It's very common on nature strips. And I suspect that part of the reason for that is that the seeds are very readily spread around the landscape on dog's fur and in our socks and bootlaces and stuff like that. To the extent that I don't know what the pre-European distribution of this species was. I mean, there are some very early records of it, and so it would have been probably natural where those specimens were collected. But for example, I lived in Montrose from 1983 through to 2002, and there was no Ritidosperma racemosum in Montrose until somewhere around 1995, because I was really focusing on wallaby grasses, and I just could not find it until I found it in the Montrose Uniting Church. Now you look anywhere around Montrose and you'll find it everywhere. So it has absolutely exploded, not just in Montrose through that period, but through Victoria generally. So and because early botanists didn't really waste a lot of time collecting grass specimens, we don't have a very good record of what the pre-European distribution of most grasses was. So whether you call it an indigenous species on any particular list is a little bit arbitrary, and rarely can it be confidently asserted. Okay, so we have identified a wallaby grass, and checking the time, if I get rid of that, I see that it's 3:45. Crikey, I'm really going to have to get going. Why did it take so much longer today, this afternoon than this morning? Okay, here's some extra homework for you. It is take that other wallaby grass stem, the one with much stumpier, or for Maddie, the less slender inflorescence, and see if you can identify it. I reckon there's a good chance you'll be able to do that, and I will tell you the identity of it next week. So I'll give you a week in which to have a go at that. Right, now I've given you another specimen, which was the needle grass, and I'm going to use that to introduce you to the key to Victorian grasses. So can you please get the notes called Key to Wild Grasses in Victoria? And I'll see how quickly I can do this, because we are running out of time. Right, I've got my specimen. Okay, The key is again a dichotomous one. Oh, I should ask, has anybody got any questions or comments before we progress? Okay, away we go. In this key, I have eliminated all the complicated couplets that you'll find in any other key to grasses as far as it's been possible. And instead I've introduced characters that I don't know of it ever being used in any other key in the English speaking world for grasses, or any other language for that. It's just that I only know about English language ones, including the growth habit. The key starts off asking you to consider the habit of your plant. Like this, 1A says the growth habit is like a reed or bamboo with creeping rhizomes or underground runners and rigid upright stems. I'll pause there. I collected these specimens. They were just tufted. I dug them out. There were no rhizomes. It doesn't look like a reed or bamboo. I could read on through the rest of this, and I should do, except that we're running short of time. So I'm going to say at this point that it's falling down already. That can't be right. 1B says the plant doesn't match some or all of the features above. I'm going to go to number 2 just to save time. 2A says the plant stems are creeping, or else they climb or scramble over other plants or objects. These stems were quite upright. The top of them were arching over a little, but they were certainly self-supporting. There's no way you would call these Chilean needlegrass specimens creeping or climbing or scrambling. It goes on, the stems produce side branches. I see no side branches. So it falls down on all of that stuff. So on the same basis as couplet 1, I'm going to say, to save time, I'll skip over the rest of that. 2B says that plant stems do not creep, climb, go to 33. So let's zip down to couplet 33. Uh-oh, there was an error processing a page. I think I'm going to have to shut that down and open it again. Recent. Okay, we're going to 33. Right, 33A says, for 1 centimetre or more above the ligule, each mature leaf is at least as thick as it is wide, comprising the strongly thickened midvein and just a trace of flat tissue fringed with soft, silky hairs. It's asking you to have a look at your leaf blade, and I really should say leaf blade rather than just the leaf, just to make it crystal clear, but we're above the ligule, so that does mean the blade. And we're looking at the space from 0 to 1 centimetre from that ligule along the leaf blade. And the edges of my leaf blade have curled in because they've been in the back of a car out at Toowoomba Vale in the heat. They were flat originally, but I'm hoping you would agree that the thickness of the leaf is not nearly as great as the width of the leaf. Having flattened my leaf blade out, it's about 3 millimetres wide and it's not 3 millimetres thick. So we have a grass that doesn't fit this description of Imperata cylindrica. Now, I do actually have a specimen of what I'm talking about, this Imperata cylindrica. I'm thinking I better take it tomorrow because we're going to run out of time otherwise. Okay, we could read on. It says, I'll skip some of that. The leaves form clusters at intervals along slender, scaly rhizomes. And I already said that this grass had no rhizomes. Then it says, inflorescences are silky, hairy from dense white hairs. The flowering is uncommon except following defoliation, e.g. by fire. Well, I wouldn't call these inflorescences silky, hairy, and I don't see these dense white hairs, so it's not Imperata cylindrica. Go to 34. 34A says the inflorescences comprise burrs or spiny packages attached closely to the central axis or rachis. Each burr has tens of hooked or minutely barbed spines, so it clings to things. And that would take us to 35, or the alternative to 36. If we jump ahead and had a look at what would happen if we went to 35, you'll see that I've got some photographs of the things that it's describing there, the burrs, and our specimen doesn't have anything like this in the inflorescence. So we won't go via 35, we'll go to 36. 36A says, within each inflorescence, not just at the base of the inflorescence, there are multiple leaves, or strictly leaf-like bracts, at least 1.5 centimetres long, right up to within 0 to 2 centimetres at the top of the inflorescence. Now, if we were to take this root and down to 41, we'd see that it has the 2 kangaroo grasses and the 4 other species that I discussed when we looked at kangaroo grass on Thursday out at Tulane Vale. And you might remember I showed you that as you look up the stem of the kangaroo grass, you've got a leaf with its sheath and a blade and a bit of stem, and then you get to where there's flowering parts. And you go a bit further up the stem into the inflorescence, and it's got more of these leaves or leafy bracts or leaf-like bracts. And no matter how far up you go, you keep finding these leaf-like bracts. So that's what you would expect with 36A. We don't have that, so we go to 37. And 37A says each spikelet or cluster of up to 3 spikelets is completely enveloped by numerous straight or feathery bristles at least 10 millimetres long. Now, in my inflorescence, I can see awns. You might think of an awn as a bristle, but this is saying that the spikelet is enveloped by them. And it goes on, the bristles are attached to the spikelet's stalk or pedicel. Okay, if you have a look in your inflorescence, you'll find that the spikelets, which comprise a pair of glumes and a single seed inside the pair of glumes, they're on their individual stalks or pedicels. And those pedicels have no hairs along them, not these 10 millimetre or more long straight or feathery bristles. So our specimens do not fit this description. I've put a photograph there that's representative of the 4 species that come out here. This one's fountain grass, it's usually sold as, and is going wild around Melbourne in particular. Because we're running out of time, I'm not going to dwell on that. Let's just say, yeah, we don't have these bristles like on one of those synchruses, and we'll go down to 38. Couplet 38 is really important. And I've taken some effort to describe in words in the lead 38A what's going on. 38A says spikelets are arranged in spikes or racemes, which may be held singly at the ends of the stems or in any other arrangement, for example, like couch or paspalum. Now, you probably don't remember exactly what a spike or a raceme is. So I've got an explanation there, and perhaps the best way to explain it to you is to stop sharing that and show you an example of a paspalum specimen. And I'll show you some other examples tomorrow. I'll get my paspalum. In this case, and I have to get rid of that microscope like that. Now you're no doubt used to seeing what a paspalum inflorescence looks like. It's got these branches that alternate. My lowest one on the right here is pointing downwards, the next one's upwards, the next one's downwards, and then there's a terminal one that's trying to go upwards. So it's got alternate branches, and each one of these branches is a raceme. And I could tell that by flexing it. Firstly, I noticed that I can see the rachis, as it's called. If it gets into focus, I'll come on. Okay, it's in focus now. We can see that the spikelets are all on the underside. That's the way it sits in the wild. And if I want to see the spikelets, I have to flex it. And when I do that, again, hoping that it's going to focus. Come on, camera. There we go. We can see the spikelets. They're oval, or they come to points, what a botanist would call ovate shape. And they're in 4 neat rows, and they come in pairs. Every second one has got a tiny stalk, and it's not getting into focus very quickly, and we don't have a lot of time to wait for it. Trust me, if you have a look at Paspalum, as we'll probably be able to do tomorrow if you want to, you will see that every second one of those little ovate shaped spikelets is on a little stalk, and the other ones have no stalk at all. The camera, go back where we were, and we've got 1 minute, goodness me. Right, we'll see if we can identify this thing at high speed. Okay, so a spikel or a raceme has got the spikelets either without any stalks or with pedicels less than 3 millimeters long, or there's a slight variation on that, which we don't have time to go into now. We'll perhaps do that either in the field tomorrow or next week. So we'll go via 38B to 84. It always gives me a sense of achievement when I zip through a whole lot of stuff. Skipping down to 84. Headed to other inflorescence types. 84A says some or all of the dispersal units or diaspores, or you could call them grass seeds, have at least one geniculate awn, perhaps as short as a few millimeters. Geniculate means it develops 1 or 2 bends as it dries. Well, we've got seeds that have clearly developed their 2 bends, so they're geniculate. And they're certainly not as short as a few millimeters. So yes, that seems all right. We could read the rest. 84B says if there are any awns on the plant, they're not geniculate. So that's not right. We're going to go to 86 at high speed. 86A says the inflorescence comprises awned seed-producing spikelets, not to be confused with florets, and unawned, more slender spikelets, alternating along the inflorescence branches. In other words, if I were to flex my inflorescence and find the spikelets along on one of the branches, I would find one that has an awn and develops a grain, followed by one that does not have an awn, one with an awn, one that does not, and so on. All of mine have got awns on. So it's not a sorghum species, one of these 2 species. We're going to 87. Do pipe up if you want me to stop, but I'm just keen to finish quickly because it is now 1 minute past 4. Okay. 87A says each spikelet produces a single floret with a single geniculate awn and 0, 2 or 4 non-geniculate awns. Well, my floret has a single geniculate awn and no non-geniculate awn, so that's a 0 bit. That was the first test that I gave you for a speargrass or a needlegrass out at Toulern Vale, wasn't it? That was step 1 in the 4-step process. So we've got the 1 seed inside each pair of glooms, whereas 87B says that each spikelet produces more than 1 floret. So we go to 88. 88A says, in addition to the geniculate awn, each floret has 4 smaller awns, 2 of which may be much smaller than the others. That would be the 5-awn speargrass. We don't have that. And this is not a dichotomy or a couplet, it's a trichotomy or a triplet. 88B says, in addition to the geniculate awn, each floret has 2 smaller awns. No, we've got none. So 88C is the one. There is only 1 awn on each floret. So it may branch into 3 arms. Well, ours doesn't, but just says it may do that. So we go to 90. And 90A says each awn divides into 3 branches that spread apart from each other on drying. and the grass grows in the far northwest of Victoria. Well, this came from Baroni, no, it came from Toowoomba Vale, so not from the far northwest. And the Orne doesn't divide into three, so it isn't Aristida contorta or Aristida holathra. So we'll go via 90B to 91. We're nearly there. 91A says most Aurora florets have awns over 10 millimetres long, as opposed to the awns are no longer than 10 millimetres. Ours are obviously much longer. In fact, I was measuring 65 to 70 millimetres this morning. So I'm going to 92. And 92A says the awn is up to 30 millimetres long and so fine that it is hair-like. That would be a dicotyledone. And that's the big difference between the spear grasses and the plume grasses or dicotyledons that look rather similar, but they have shorter awns and they are fine like hair. They also differ in having a glume with no veins other than the midvein. So we don't have a plume grass, we go to 93. 93A says the base of the floret comes to a fine sharp point, which may be almost straight or curved. And it warns you that it may not feel sharp if the floret is immature and soft, but these have dried out and they've hardened, so you'll prick your skin with them, I reckon. It goes on, the awn is 20 to 130 millimetres long. Ours are in the middle of that range, so that's good. 93B fails both because of the absence of a sharp point from 93B and that the awn can only be up to 35 millimetres. So we go to 96, skipping over this Amelicloa, which used to be called Stiper, and a couple of Nacellas. Now, 96A, well, in 96 we see we've either got a Nacella or an Ostrostiper. And so this is the fourth step in that four-step process that I gave you out at Toowoomba Vale for a speargrass or a needlegrass. And you remember how to distinguish them. It's by the corona. So 96A says the base of the lemma awn is surrounded by a collar or corona on the lemma apex, on top of the lemma, measuring 1 millimetre or more long. And you could measure that and you'll confirm that. There are hairs attached along the lemma callus, the narrow pointed section, but the body of the lemma, i.e. the cylindrical part that houses the grain, is hairless or has a line of hairs extending along a ridge. Now, if you're good with your hand lens, you'll see that line of hairs that extends along a ridge. And so this is Nacella. It's not the two above, but that still leaves 4 or 5 species in Victoria that we could have. And that's as far as we can go with this. Then we would be going to the key to stipoid grasses. And perhaps I'll leave that as an exercise for you to do between now and next Wednesday. I don't know how I found time to do that this morning. We did actually get to the individual species, but give it a go yourself. It won't take too long, I don't think, and you'll get the additional satisfaction of having done it yourself. I think you'll get down to the individual species. At 6 minutes past 4, we're a little over time. I'm sorry about that. Are there any questions or comments?
Anything we should be bringing tomorrow, Graham?
Fine, I'll deal with that. That's what I was going to do next. But are there any questions or comments on the use of the keys?
Just one. I'm not sure if I have the keys to the grasses and stipoid species. Were they in an email somewhere?
Yes, there were some links and I sent that yesterday, I think it was. Yesterday? Yeah, yesterday.
Okay,
Didn't
thanks.
get those either.
Hmm?
I didn't get that email either.
You might not have noticed, but it was in the link to the Zoom. It was below it.
Yeah, I didn't get that.
Yeah. Okay.
I just used the same link from last week.
Yeah, me too.
Okay, it definitely went out. I'd suggest checking your spam filter, because if I send it again and it's gone to spam the first time, we'll do the same this time. What can I do? I can put in chat. No, I can't really, because it's such a long lot of text for both of them. I'll try sending it again. So the people who missed it were Katie and Emma. Anybody else not get it? Kelvin? All right. I will,
Not in
and
my
Katie,
spam folder,
pardon?
and it's not in my spam folder.
That's interesting. I will send it again in a few minutes. If you don't get it within the hour, can you let me know? And I will come up with a different way of doing it. If I send you a text message with the link, that might be more reliable. Okay, so I'm doing Kelvin, Katie, Kate and Emma. Is that right? Yep, okay. I'll do that. Now for tomorrow, I suggest you bring a raincoat for number one, because if we all have raincoats, it won't rain. That's the way rain works, isn't it? We shouldn't get much rain. Certainly not as much as came down last time. Gee, I'm glad we got back to the cars when we did. I would recommend bringing the checklist to plants at the Montrose site, which I will also send you. Well, now those 4 of you who didn't get the notes from yesterday's email, I'll send it. It might not be within the hour, but it'll be within an hour and a half, because I'm going to compile a checklist and I'll send you that checklist plus the links to the notes. So bring the checklist, bring the key to wallaby grasses, the key to stipoid grasses, something to eat and drink, and I think that would be about it. And you'll be going to the end of Kirkwood Court in Montrose. You've all got a link to a Google map that will show you where to go. There's a dead end at the end of Kirkwood Court, and you'll see that there's a bushland reserve called Lavertia Reserve. You just walk up through the gate there to the top of the hill, about another 30 metres away, and there's a picnic table on the top of the hill, and we will meet there. That's all I can think of about what to take. Maddie, has anybody got any other questions or comments? No, then it's class dismissed. I will see you at 1:30 at Montrose tomorrow. See you later.
Thanks, Graham. Thanks, everyone.
Thanks, Graham.